Citrus is an integral part of California's history and culture. Considering how we even have a whole county named after the orange, it's not surprising. California has been one of US's top citrus providers for over 100 years. In fact, since 2016, California has been the top US producer of oranges. However, the forecast of oranges has not been looking too juicy. Huang, a disease responsible for the deaths of billions of citrus trees across the globe, is gradually spreading through California. But what exactly is going on, and how can we use food science to stop this? Huang, otherwise known as citrus greening disease, is caused by bacteria from the genus Candidatus liberibacter. While there are several members of this family that causes HLB, the one with the greatest global impact is Candidatus liberibacter asiaticus, often abbreviated as CLAS. So, how does CLAS infect trees? CLAS must first get into the tree's phloem. This fluid transports nutrients from leaves to the rest of the tree. It's kind of like our blood. Without the optimal conditions, CLAS will not thrive. But once CLAS enters the phloem, it's in paradise. It has plenty of food, the perfect temperature, and no competition. As CLAS moves through the plant with the phloem, it consumes plant nutrients and secretions of various molecules, depleting food from the plant to thrive. Plus, if the bacteria reaches the root system, it has access to consume stored nutrients. Meanwhile, the secretions from the bacteria can negatively affect plant development and suppress plant defenses by killing cells. So what does a diseased citrus tree look like? The tree's leaves will become yellow and blotchy, and its foliage will be sparse. Its fruit might drop early. Even if the fruit survives long enough to ripen, it's usually green, lopsided, and smaller than average. The resulting product will be bitter with dark seeds inside, making it unsellable for the market. Almost all major citrus growing regions across the world are infected by Huang. What happened in Florida, another major citrus provider in the US, shows the dangers of this disease. In August 2005, Huang disease was found in South Miami-Dade County. Many farmers didn't take proper prevention methods, such as culling fields that had an infection. This promoted the rapid spreading of the disease. By 2015, HLB was confirmed in all of Florida's citrus-producing counties. Today, over 90% of all citrus trees in Florida are believed to be affected. Citrus-bearing acres have decreased from around 680,000 to 390,000 in 15 years. Orange production has gone from 300 million boxes in 2000 to 70 million boxes in 2019. All this and more have contributed to a loss of billions of dollars. But why hasn't anyone been able to control it? After all, it's just a tiny bacteria. First, infection isn't always obvious. CLAS can travel through the system, causing minimal effects up to years. By the time symptoms do start showing, the infection runs deep. Second, CLAS is hard to study. It needs the perfect conditions that resemble phloem to survive and thrive. Scientists only recently discovered how to grow the bacteria for long periods of time in a lab. So if CLAS can only live in highly specific environments, how is it getting around? There's another nutrient-rich environment in town: insects. Specifically, the Asian citrus psyllids, or ACP. ACP are highly invasive pests that feed on the leaves of citrus plants. Doing so, they can pick up CLAS and bring it to another plant. In fact, this relationship is what makes HLB a large-scale problem. All you need are a few HLB-positive psyllids to be moved to a healthy grove. The ACP feasts on the leaves, and CLAS enters the plant. As both parties reproduce, the cycle continues rapidly until an entire grove of trees get infected. Various treatments have been tested: antibiotics, pesticides, thermotherapy, and parasitic wasps, none of which are fully effective. Amongst these, California has been relying mainly on pesticides. It has helped, but it's still not enough. This all seems hopeless. Is there any way to save our citrus trees? Fortunately, food science is giving us the solutions we need. Research has gone towards solving this problem in 3 ways: changing the tree, attacking the bug, and killing the bacteria. Let's take a look at some of the innovations in each category. Starting with changing the tree, some plants, such as sugar-bell, have been found to be more resistant to HLB. Some scientists are breeding these resistant cultivars with citrus we know and love to see if they can introduce the resistance genes into commercial varieties. Other scientists are using CRISPR to change the citrus trees. These scientists first look for which genes are being affected by the secretions from the bacteria and modify it accordingly. If the bacteria can't affect the gene, it has a reduced effect on the plant. A major limitation to both of these solutions, however, is that trees would have to be replanted. That's an expensive loss. What if we attack the insect vector instead? One research group has been using a technique called RNA interference to mutate insects. The presence of a certain kind of RNA will shut down the production in the corresponding gene, so scientists have been studying and developing RNA molecules that trigger the destruction of genes working in pesticide digestion or wing development. Another research group has been studying how to use the citrus tristeza virus, or CTV, against insects. This virus is a pathogen that infects citrus phloem. However, this CTV strain is modified so that it is milder and so that it carries compounds that poison psyllids. CTV is then injected into the tree, and when insects take up the CTV-filled phloem, they die. But recently, there has been a bigger breakthrough for killing sea lice. In July 2020, a research group in Riverside, California, announced that they found an antimicrobial peptide that cured HLB. The AMP is derived from the Australian finger lime. After the AMP was injected into the infected trees, researchers observed a drop in symptoms and bacterial count. The compound is safe for human consumption, easy to make, and stable at high heats up to 54 degrees Celsius. It is suspected that the AMP could even be used to vaccinate younger trees. Field testing and approval still need to occur, and the long-term effectiveness has yet to be studied, but this substance is being hailed as the first substance capable of fully controlling HLB. This wouldn't just save California's citrus economy, it would affect the world. In application, we would most likely have to use a mix of solutions, attacking the disease from more than one perspective. For example, using wasps to kill ACP and inoculating trees against sea lice. Additionally, we must continue with prevention methods. Even if we have treatments, removing infected trees and reducing ACP counts are both key in preventing HLB from spiraling out of control. California is the home to millions of US's citrus. HLB is a tough problem to attack, and time is not on our side. California's future of our citrus lies in the implementation of our current tools and continued innovation in the field of food science.